It's my great pleasure to introduce Francisco Balzarotti. He just won the Frontiers of Science Award, 14th of July, in the Great Hall of the People in Beijing. And he also has a huge Wikipedia page, so it's huge. It's huge. So I'm cheating, but you got your PhD in University of Buenos Aires in electrical engineering, in nanophotonics, nanoplasmonics, super lenses, nanolithography. That's a good, yeah. And then you moved to Göttingen in the Department of Nanobiophotonics, which Stefan Hell, where he developed the MINFLUX technique that you're going to talk about today, right? Since 2020 was a great year to start a lab, right? He's a group leader at IMP in Vienna. He leads an interdisciplinary group of microscopy and biophysics, and I can't wait to hear everything about it. Thanks so much for being here.
Thank you. Thanks for the very nice introduction of history of what I've done. So I'm thrilled to be here in person. It's great that we could work that out, and it's great to visit Janilia. It's very refreshing to talk to colleagues of neighboring or the same area. So yeah, that it's a great honor, and also it's great to have it mirrored in Heidelberg. So now today I'm going to be talking about MINFLUX. I will give you some insights into my understanding of the reasons why it works and what is the actual source of the information. I would like to pass on this message. And also I will be talking about our developments and technology and some other tangents we have been taking lately. So yeah, let's see how it goes. So I call this talk Accessing Nanoscale Structure and Dynamics with Light, because many of these light-based techniques can nowadays reach precisions of localizations that are in the nanometer range and so on. But if we think generally, which roles could light microscopy be fulfilling for biology? You could extract in situ, so in cells, stoichiometry and conformation, even if you have enough resolution and sampling, labeling, and also dynamics in vitro. This is something we're doing now, but also in cells. And there's some papers out there that are getting into extracting conformational changes in cells. But for this to be done, there are a set of key requirements, which is you need to have targeted labeling on specific, very precise and very complete high efficiencies of your fluorophores. You need high localization precision. You need isotropy. You need high temporal resolution or appropriate temporal resolution for what you're looking at, and multicolor multiplexing capabilities. You need to look at many things simultaneously. So the history lesson, you may have seen this many times. Light microscopy is limited in resolution, but still is still used in spite that the electron microscope reaches way better for decades in terms of resolution, but we use it for specificity, contrast, and light compatibility. This limit was written, or a hint of this limit was written in stone in Germany, Jena, many years ago. In the mid-20th century, there was a proposal to a window to the nanoscale through far-field readout. This is forced resonant energy transfer, and a lot has been done with that, dynamics and so on, but it's not that you make images with it. And finally, the say true far-field super resolution techniques were proposed in the mid-90s and led to the Nobel Prize in 2014. Everybody here must be very aware of this. And part of my work while I was with Stefan was, okay, how far can we take this? Now, this arrow is a bit metaphorical. It's not that you want to go to the picometer, but what are the limits? In terms of spatial resolution, temporal resolution, where can we go? As you may know, there's a lot of techniques that try to fix the shortcomings of the confocal or the wide-field microscope. This is absolutely arbitrary and incomplete. And you may want to think when you're classifying these techniques you may care more about spatial resolution, temporal resolution, or toxicity or whatever. So this is arbitrary. But there's a family tree of ever-growing techniques. Now I think this has slowed down. There's not an exponential growth anymore of techniques. And today I'm going to be focusing on single molecule localization strategies and the MINFLUX localization in more detail. So to give you a sense of scale, this is a ubiquitin molecule, and this is a dye that would be labeling it. And if this ubiquitin molecule was in a chain, targeted labeling, marking a substrate for degradation, and you wanted to look at this, well, you would only see these few dots. This is what you have to do. And but this is quite small. This entire field of view fits in that fraction of a pixel. So that's what the challenge itself. And of course, you cannot distinguish them. So what was proposed many years ago is to have them alternate blinking, or have their emission being alternated, and localize each one of them one at a time. And the precision of localization on that case scales with wavelength, numerical aperture, and number of photons. The more photons, the better your signal, the better your fit to a model of this thing, and the better precision you have on the parameters that you extract. That's why more photons give you more. Now, if you get for such a challenging object, a 10 nanometer sigma localization would look like this. If you get more photons, you get better and better and better. And at some point, you could reach something that might look like that. But you have this issue down here. Sorry, I'm not using the pointer for people online. You have this issue here with the 10,000. So the MINFLUX localization can reduce roughly, or up to an order of magnitude, maybe more, in the requirements for photons for localization. And that is because the dependence of the precision doesn't have at first order the wavelength or the numerical aperture. It has a geometrical parameter that you can tune, and you can shrink quite a lot. Not to zero, but quite a lot. And additionally, depending on how you do the experiment, you can have an exponent in the number of photons, which is another manifestation that you're boosting the information. So this is, I will dive into details about that. A message on in our field, we talk about this number, sigma, a lot. And this is just a parameter of a probability distribution. But if you have a 1D, 2D, or 3D, a sphere or a circle with this diameter represents less and less and less of that distribution. For 3D, this represents 4% of the distribution, while for 1D, it represents 38%. So you need to be aware that this number, when I say 1 nanometer sigma, and I'm talking about 3D, what I'm actually saying is that there's a 3 nanometer sphere that will host 50% of the distribution, or a 5 nanometer sphere that will host 90% of the distribution. So you need to keep this image in your head. But we keep reporting sigma. It's just that you need to be aware what this is. Very important. So how does MINFLUX work? What is it based on? So instead of localizing using the emitted or the diffraction pattern of the emitted fluorescence on a camera, you just sequentially probe different locations with a beam that has a structure. So in this case, if I wanted to observe this molecule up here, I would illuminate it sequentially with these patterns, and depending how much light I get on each one, I can triangulate where it is. This is what it's based on. And in terms of performance, what it can do, if you look at precision versus number of photons in log scale, what you have here is the a camera limit of precision, so with a perfect camera, no noise, nothing, so very ideal. And down here you have a MINFLUX experimental precisions. It can go, in this case, 5-fold better in precision for the same budget of photons, or 20-fold better, 20-fold less, cheaper in photons for achieving the same precision. Now, let's get a bit more detail. Oh no, I'm getting messages. Okay, you don't see it, great. So let's say you want to localize that molecule. So I said, you probe it with a beam that has structure, you move this beam somewhere else, you probe again, you have 2 measurements. These 2 measurements, depending on what the molecule is, it may give you more or less photons, and you should be able to pinpoint where the molecule is. So a way to think about this is as if there was a ruler. This is an analogy, but it's also quite true. Where the size of the ruler is the distance between the zeros of the beams, and the number of photons is the number of divisions. Because if your molecule was here, you would get 0 photons first and all the rest later. If you were here, you get the inverted case. And if you were in the middle, you get the 50-50 case. So each piece of space gets labeled with a photon combination that you can measure. If you use more photons, in this scheme, in this setting, you get them distributed more finely. So one combination corresponds to a smaller piece of space, and that's where the precision, higher precision, is another interpretation of where the precision comes from. In reality, this is not equidistributed spatially, but it's still true. Now, the trick in MINFLUX is you keep using few photons, but you shrink the ruler by putting these two beams that are sequentially sent to your sample closer together. So it's something that is totally controllable and doable. And there's no limit to it. You can put them as close as you want, right? There's no limit. The beam is still huge. So getting a bit more strict, each photon emission follows a Poissonian distribution whose mean is proportional to the intensity that is exciting. And to localize, you can look at this sensitivity function, which is a fraction of photons you collect in one of them, which will be proportional to the ratio of intensities. And this number, it's a function that goes from 1 to 0, spans full range from 1, 0 to the next 0. And this is the key where the trick comes from. So let's say that you happen to measure 60 and 40 photons. This number is 0.6. So you go to this function that you know very well because you know your beam shape, and you find x. That's all you have to do, and that's a maximum likelihood estimator, by the way. So if you look at the distribution, now these numbers still comes from random distribution. So it will have a distribution itself, this p. And going through this function to the x-axis is what gives you your localization. So mapping this distribution to the spatial axis will tell you what is your distribution for localizations. And whenever you're devising a new method to measure something you may care about is how precise it is. Is it better? Is it worse? There's a way to check how small this distribution can be, which is looking at the Kramer-Rao lower bound. This tells you what is the best possible precision or the minimal standard deviation that a maximum likelihood estimator will achieve. This would be concretely it. And to compute that, basically this information is the inverse of the variance, by the way. So the more information, the smaller the variance. And it looks a bit scary because it's the average curvature of the log likelihood function of the problem. It's a bit of a mouthful, but it's not that terrible. What you need to plug in there is your Poissonian statistics and your geometry. And if you solve it for a situation with quadratic beams, so quadratic approximation of these beams with the parabolas, this gives you a very innocent parabolic function. So super, it's really one page, not a big deal. And when you evaluate this at the origin, surprise, surprise, in comparison to Storm or Stead, you get this L there. This L is the distance between the two beams, is this ruler that I'm showing up here. And this tells you that, well, you can use more photons to get more precise, or you can just shrink that number. And an interpretation of that is that as you shrink the distance between the beams, this function that spans full range from 1 to 0 spatially, and is mapping the photon distributions to the spatial axis, gets steeper and steeper. And that's why this mapping here becomes narrower and narrower. This is where the precision comes from. The Kramer-Rao bound, if you plot it, represents this, but I think this picture is more important because it really tells you, how what is this. So we did call this MINFLUX. One way to think of this acronym is that the M of MINFLUX is maximally informative luminescence excitation. So we're trying to maximize the visual information that you extract from certain parameters through patterning the excitation. Something curious and nice is that everything that is in meters, like this or this, gets adimensionalized by this pattern, by this parameter L. So this means that if you shrink the distance between the beams, zoom in and increase the power of your exposure time, and keep doing this over and over and over, as long as there's no background, the function that is doing the magic, that is doing the mapping of photon statistics to localization statistics, is invariant to spatial scalings. This invariance to spatial scalings is what gives you the magic. So all in all, the precision scales with that thing up there, with L, and you can touch L as you want. So for localization still, you need to have blinking, as in other super resolution methods. So you get your events. One oddity of MINFLUX is that we could have adaptive activation. So we will photoactivate. When a molecule appears, I stop activating. and then when the molecule goes away, I activate again. So with that, you can avoid collisions and double activations. And if you analyze each one of these events, for instance, this event has the yellow donut the lowest, while this one has the blue one. So this tells you that this molecule is closer to the blue, and this is closer to the yellow location. Still, remember, the donut is enormous. So if you do that for DNA origami, you can cluster and so on, but even without clustering, you can know which molecule is which, because the sigmas of the 2D distributions are in the 1 nanometer range, plus minus something. So with 500 photons, you can get already there. Now, if your molecule moves and you get a trace of photons, you can transform it into a trajectory to the tracking. And when things move a lot in the, sorry, not a lot. When things move in the very small scale, well, they will fit in your pattern of zeros, and you can track them. So we did this with origamis, flipping origamis, and we could reach 2 nanometers with a sub-millisecond precisions, few photons. Still, this is a very high emission rate. This is 300 kilohertz of count. So you need a die that is kicking to do that. Otherwise, so you need photons. Right? And finally, if your molecule gets out of your ruler, well, you can put a feedback loop and track it around, of course. And this is something we did to track the small and large ribosomal subunit proteins. And for the photon budget that photoconverter proteins give you, this was in live E. coli with agarose pads, super noisy situation, you would get a mean mean number of frames if you're tracking with a camera of 10 plus minus something frames. But the number of actual localization with MINFLUX for the same budget was 700, so 700 localizations or so, because we were using roughly 10 photons per localization at even higher, 8 kilohertz. So, in this case, we push for speed and not for high precision of localization. So, all in all, there are many strategies that you can use for this. So laterally, as I show you that, it doesn't have to be a triangle, it can be anything. If you have a beam that has a 0 in the actual direction, you can displace your beam up and down. I will be talking about that in a bit. And you can, your Z, your actual localization precision will be decoupled from the XY. You can do whatever you want with it, which is great. And also there's an iterative version of this where you can use some photons with some set of beams and pattern, localize, then shrink, localize again, shrink, and shrink. And when you do that, what happens is that the number of iterations that you're doing, if you're doing the optimal ones, ends up in an exponent in the number of photons. This is a very curious thing, but it's just another manifestation of the higher information. You're jumping from, so in localization versus number of photons, you're jumping from one line to the next, to the next, to the next line. This is what's going on. I have more, if you want, in the questions, we can discuss that. Now, if you want to do images in cells, this is still a bit tedious because you need to do some sort of mosaic. So you go somewhere, you photoactivate, then you do this iterative procedure very quickly. The raw data looks like this. So you have nothing, then a molecule is photoactivated, or comes, binds, right? It could be paint, it could be anything. And then you have the iterative process happening in a few milliseconds. And then with the smallest pattern that makes sense to use, you squeeze out everything that remains. That's the procedure. So do this at each location, and then you end up having nice images. So these are nuclear pore complexes, the label with the snap tag and Alexa fluorescence 47N, doing storm with the blinking buffers. In this type of image, in 2D, we got median localization precisions of 1 nanometer, meaning half of them are better. It's very challenging to render these things, and the Fourier ring correlation for 2D was touching that point. Whatever, I find this a bit arbitrary, but I report what people like to see. You can also do this with photoactivation, of course, of fluorescent proteins. This is a comparison of classical wide field palm, and this is MINFLUX palm. Of course, the proteins are less bright. There's more background and so on. Or worse signature background ratio, or less photons in total. So the precisions are a bit worse. But the cool thing about this is that you can do it live. So if you go to a live cell and do this in a small regions, right? Because it takes a while to do the whole mosaic. But if you do it in a small region, you can get these images in 20 seconds or so. And you can push it further. We really didn't push it to the limit. As I said, it's a bit challenging to render this data fairly, because so we render this with some kernels, but the actual raw data that corresponds to this is this. So you can see that here you see this overlapping localization, but we were very conservative in the way we were rendering this data, because we're using, say, these circles, these ellipses that you see here, that I think these are 2 or 3 sigma diameter ellipses, but you see that the clusters are absolutely distinguished from each other. So very challenging to do justice to the quality of the data. Another thing to notice is this is DNA origamis in 3D, and they're never flat, by the way, at this level. But this is storm MINFLUX imaging. Something that you should know is that, okay, you get these distributions and so on because you repeatedly localize, but then you can also take all of them together, get the mean, and report the standard deviation of the mean. And with regular storm, this is always in the sub-nanometer range. Now, what's their drift? Do you care about this number at all? The labeling distances, that's another story. I'm just talking about the performance of the localization procedure itself. So as I said, 3D worked well. We did this on nuclear pores. And funnily enough, the axial precisions were better than the lateral ones, because we made some mistakes on the axial calibrations. So then we put a smaller pattern in XY than in Z than in XY. And something interesting, this is perhaps what I intended to say before. So you have the precision versus number of photons. This follows in log. This has a -1/2 slope because it follows the square root. Follows the square root of the number of photons. Now, when you do one iteration and another and another and another with different L's, you're jumping from one curve to the next, And this is why you go steeper than the square root dependence. What's interesting here is that there's this quantum camera bound that tells you, how precise you could be if you forget a bit about how you integrate in space and in time the PSF. It's a bit of a more strict and general bound. And you can cross this in the lateral and the axial direction, whatever. It's another manifestation of the same thing. We do need to take care of the shapes of the beam. This is all based on knowing the shape for real. So we do use adaptive optics, and we perfect our donut. To begin with, this is okay, but then it needs to get better so you can have full control. And especially, oh, I'm missing a slide here. And especially, let's see, because I want to show you how bad it is for the other beam. So this is the same system. You put a top hat beam. That's what it looks like. So with these corrections, you can really get it nice. This is very important. Otherwise, your localizations are going to be biased. Multicolor can be done as it's done for many other techniques. You can just put split your detectors and use dyes with different spectral shifts for identifying them. So you can add all the counts to localize and do classical MINFLUX. And then you can just use the spectral ratios to classify. These populations are normally extremely separated from each other, and put colors to it and do 3D multicolor. That's not, it has to be done, but it's not so terrible. And note on the fluorophore interactions, which is something that, sorry, it's something that comes up a lot. They say, well, what if they're very close to each other? What happens? So when you have your fluorophores closer than 5 nanometers or so, even though you stop activating, you get these events with multiple fluorophores being activated anyway. So here you have a zoom in of that. So you have the purple fluorophore up here that it's active for a while. Then the red one gets activated, and then the green one. It's always the neighbors that activate each other. And you see this only when you have 5 nanometers or less separation between them. In my experience, when you're labeling real stuff, you're never in this range. Even we try to label actin monomers to look at all the components of the actin filaments, and even there, the predicted distances are 8 nanometers. So, we didn't see this at those distances already. But it's curious, and if you wanted to study some weird interactions, sure, this could be a way. And then you can see each one. And of course, when they're both overlapping, you could use a Bayesian approach and use the neighboring information to refine everything with the photons where both of them are emitting. So a note on the beam shapes, as long as you have two zeros close to each other, this will always look like parabolas. So the P function that maps the statistics is the same. The Kramer-Rao bound goes lower and lower. And note on that with Gaussian beams, the more separated they are, the lower the Kramer-Rao bound. Okay, and you can do all of this with Gaussian beams as well. And the precision scales with the sigma of the Gaussian PSF squared divided L. So it's in the denominator. Pretty funny. But that's not what I want to talk about. So something that received attention is standing waves, because one thing is that you get so spatially uniform resolution or precision, sorry, and you get a factor of 2 or so better than regular camera localization if you're doing SIM on top of localization. And a lot of groups, so there was a hint of this earlier on, but a lot of groups jumped into this. There were like 4 papers, and all in all, it works. So you go from something that looks like this to something that looks like this because you're getting a factor of 2. But a factor of 2 is a lot. It's not nothing. It's as if you were getting 4 times more photons from your dye. And if Luke or anybody comes up with a dye, gives you 4 times more photons, everybody will jump and use only that dye. So it is something valuable, only that it looks like a factor of 2. So all in all, to summarize what has been happening with, the initial super resolution techniques had their dependence on their precision or resolution, if we talk about STED. With MINFLUX, there was a bit of a new ingredient that seemed very interesting. SIM and SMLM has been combined, but also iterative MINFLUX came right in the middle of that. And MinSTED also, which is a sequential localization strategy with STED beams. You just sequentially localize with STED. And you're using Gaussian effective PSF that are narrower than the diffraction limited excitation PSF. Then I will make some extra comments. So in summary, MINFLUX is a localization strategy. It is sequential. It's a sequential measurement and it's ratiometric. And or if you want, you're using a set of finite points of a smart choice of a PSF of your system. This is what you're doing. It's not a tracking strategy because the role of this localization strategy is to just tell you where the molecule is. Then the tracking will be done by a feedback controller, which is an entire field of electronics. You could do optimal control. You can do all sorts of robustness and tricks, non-linear tricks to get it better. And that's a whole other side of the story. So that's very important to separate, I think, in my opinion. So it's not power intensive. You put 3 to 5 times what you put in a confocal microscope. So it's not terrible at all. And as long as you don't consider what you put on the confocal terrible. And it's not easy to build because you want a lot of elements here. And it's not trivial to process as well. There's a lot of work there. The minimally necessary elements to play this trick, to play this manipulation of the information, is to have a spatial gradient of the mean of the thing you're measuring and a spatial gradient of the variance of the thing you're measuring. So in a nutshell, if the thing that you're measuring was not photons, and this was not Poissonianly distributed, this was, let's say, a Gaussian distribution, okay? And this Gaussian thing that you measure had its mean spatially dependent with a parabola, but its noise was not dependent spatially. This all falls apart. This does not work. You need and the Poissonian distribution gives you that. Of course, the mean and the variance go together, so both are varying spatially. That's where the trick comes from. Without that, it's all forget it. And then non-necessary elements to call the thing MINFLUX. So it doesn't have to be fast, though it's nice to have it fast. It doesn't have to go to a single nanometer, as I showed for the tracking. It doesn't have to be single molecule. You can have a small object, and this would be fine as well. It doesn't have to be fluorescence. All these tricks work for elastic scattering. Things with elastic scattering, you get millions of photons. So do you care about this? Maybe, depends, the speed. I don't know. I'm waiting for somebody to jump into that. Would be interesting. It doesn't have to be a triangle pattern. It can be any pattern. What matters is that this is constrained within a finite distance, basically within a small region. And finally, you don't need a zero, because these tricks of manipulation of the information also work with Gaussian beams. The things, they're not the best choice, but they are true. With the donut beams, it is better. And also it's much easier to know the shape of a beam around the zero, because the diffraction limit forces it to be a parabola, than to know the shape of a beam of the decaying tail of a Gaussian, which, you know, anything could happen. So that's part of the choice. Now, how are we doing? We're fine. I think that one of the things that can be used, one type of object that you can attack with this technique is flexible and homogeneous and dynamic objects that are not easy or possible at all to be studied with this without techniques. For example, we are diving a bit into looking at conformational changes of SMC complexes. These are complexes that maintain the structure of chromosomes. One of them is cohesin, which has been shown in vitro to extrude DNA. This is DNA labeled, and this is a cohesin molecule. Going through it, it is kind of like a ring, and it extrudes the DNA inside of this ring that you see there. And this is kind of a large object, quite flexible. So it's beyond the range of threat. So, and it has more degrees of freedom than one. So if you really want to know what these objects are doing, there are many examples of objects of this kind, you need more. And on the other side, even for things that are static, for example, here what you see is RNA particles, mRNA particles that are made out of RNA and a bunch of proteins. This is a tomogram, so 3D tomograms, and when you average them, you can get beautiful reconstructions of some of the complexes that are there. But for the RNA, you get nothing. But it's very unlikely that the RNA is just a soup of noodles, because there are many proteins, for instance, tagging the splicing sites that are clustering together. So there is a suspicion and hypothesis of a hierarchical structure there. But how do you study this? You cannot do EM, you cannot do particle average. So, you know, tracing RNA at the nanoscale could be one way. Now, this is an optics interest group, so I'm gonna be a bit harder on the optics. So normally we put these silly cartoons in the papers, but that's not true. Then we put this other silly cartoon in the SI. This is one MINFLUX microscope, so it's composed of, it's a confocal microscope that has fast steering, beam shaping, you have wide field, you have photon counting, and you have stabilization. These are the elements. And of course, it could have less stuff, but if you want all the perks, it becomes big, basically. And why wouldn't you put all the perks? So it takes an entire table, more or less, and what you see down there are the parts to build one of these. A note on the stabilization, which I never talk about this much, but I think it's important. Our stabilization is a TIRF beam looking at in Z at the displacements of the sample with respect to the objective. That's what you see up here, and this will move laterally when this moves in Z. And then these are dark field infrared fiducial gold fiducial beads. And what you see here is 10 nanometers peak to peak of these traces over here. This is us playing music. So, you know, this should be a silent lab, sadly. And this is glancing slowly with your finger, doing this on the table. You know, you just do that and then boom, everything is moving. Then if you really push it, you see more. So it is required to have a system of this kind running active stabilization, because we cannot play RCC corrections, because when you do the mosaic imaging slowly, you know, you're probing one place at a time. So the drift, you cannot get it from the data. So that's a little detail. Now, going back to the silly cartoon, something that I said, so I do care about doing everything in 3D, because I think a fraction of the problems that are out there can be attacked with 2D imaging, but I think many of these conformational changes, the RNA particles, they need 3D. And 3D has been a nightmare because we use an electro-optical lens, which is a very temperamental device, very expensive, you can break it very easily, basically. It's terrible. I can go into detail about it. It's a great device, but very experimental, not so robust. You can go with a deformable mirror, but still you have, it's mildly expensive, and you have it's not that fast either. So we went for a solution where we just use an electro-optical modulator and a lambda quarter. And it's about the following thing. So if you take any beam that has a 0 of intensity, what will actually happen is that the electric field crosses from negative to positive around the 0. So if you manage to interferometrically combine this with a constant beam, the result of this will be displacing the 0 of intensity in whatever direction you're looking at. So this is the situation for a vortex beam across the lateral direction, and for a top hat beam across the axial direction. So if you actually overlap, this is the axial direction of the top hat beam, and this is the Gaussian beam. If you overlap them interferometrically, you can actually have a very decent performance in displacing these beams. So this is something that, and what you gain from this, well, is you're translating an amplitude modulation to a displacement. And if you can modulate the amplitude fast, you can displace fast. So we did this. And the way we did it is with a polarization interferometer. So what we got is some linearly polarized beam. We can rotate it with an EOM, so we can put some energy on the orthogonal direction. And then with a double pass configuration on SLM, we would pattern first one polarization, then image the SLM onto itself and rotate the polarization 90 degrees with a lambda quarter double pass, and then pattern the second polarization on the other, on the second pass. And finally, what you get is two beams that are orthogonal and arbitrarily patterned. And finally, what you can do is rotate 45 degrees and put a polarizer. And this is where the interferometer ends. So the interferometer starts at the EOM and ends at the PBS, very far away from the sample. You can make it as compact as possible. And finally, yeah, that's the result. So this works, and we had, you can have a 600 nanometer range or so, which is beyond what we need for MINFLUX. The nice part is using the SLM. The double pass kind of looks like this. So, you go there, so you go in, you pattern, you go out, you go through a lambda quarter and a mirror and a lens, and go back and capture the beam out. So with this, we could do MINFLUX with different beam shapes, Gaussian vortexes, displaced top hats. What you see here is this displaced top hat, step response of few microseconds. And each PSF here is measured every 400 nanoseconds. So this is not so easy to do, but we managed it. This is beautiful. And of course, there's a lot of real estate on the SLM. So why not? We can use more. So what we actually built is a 4-channel interferometer, where each channel can have 2 beam shapes that can be interferometrically combined. You can put any color and any channel. So we made 2 channels for the green, 2 channels for the red, so we can do all the shapes with all the colors and all the displacements. This is nice. Also, something interesting is that there's an algebra of combining beams. So you can put a vortex and a Gaussian, you displace the vortex. You can put a half moon and a Gaussian linear polarized, you displace the half moon. You can have, you can create triple zeros, double zeros. You can have tilt planes in the axial. You can do a lot of stuff, and this will come handy in the near future for many things we have at the store. So you can track this, and our typical object to test this is DNA origami that are standing vertical, sorry. And we have some oscillators that have a die on single-stranded end that can hybridize to another piece of single-stranded DNA that is coming out. And these are short enough. These are the paint type of DNA paint strands. So we can have nice oscillations, very controlled. And also we push this in speed. So we could actually even do this in the 50 microsecond range or so. So very, very fast if you care about it and if you have an application for it. Some other technology explorations that we have are looking at, you can put different beam shapes up there. So what is the best beam shape? Can you mix beam shapes? What will happen now that we can create any shape there very fast? This is something we're studying. And of course, on the detection as well, you can have spectral channels, you can have lifetime beams, and you can have spatial beams with detectors that are arrays. So, and the interesting thing is that you go from 4 measurements, 4 number of photons that I was showing before, to hundreds of, so it's a very sparse measurement. And in this sparsity, there's a lot of information about the background levels. So we have modeled this. We made packages in Python. We have a vector field simulator. We're closing loops with PyTorch optimizations. Our packages also have an estimator that is part of a pipeline from our data acquisition to a final image visualization and so on. So this is something that will be coming out later on. But an example that I can give you very simple, this also shows you the real scales of the MINFLUX exposures. What are they doing? So if you do 7 top hat beams versus 3 top hat for vortex, if you compare these 2 scenarios, the yellow is the second one, you just can get more precise, all in all, in X, in Y, and Z. And this is more important as you shrink more and more. So mixing shapes gives you a lot. Something that matters a lot is the background, because at the end of the day, when you have background, you'd end up overlapping 2 things that don't have a 0, it's just flat. And that's what kills everything. And different shapes of excitation gives you different background blobs on your detector. And they're very different from each other. It's not the greatest in our curve, but it kind of goes with our model. We have a model for volumetric background generation, and this is something we're looking into. So the Kramerov bounds, if you go from all top hats to mixed beams, become what you see here is all sorts of plane slices. And in all cases, or in a lot of cases, you get better performance further away from the origin. And if you mix in there spectral information, if you know that the spectrum of your dye, or if you know the lifetime of your dye, this becomes better and better and better. So the more you put there sparsely, the better it goes. And this is an example of, for example, the action. So this is a simulation, but ZX localizations for molecules that would be spaced every 10 nanometers with the mixed beams, and with the mixed beams plus the lifetime. And you see that you can squeeze more and more and more. Also with spot arrays, if you go to a spot array that is ideal, or as ideal as a single detector, is great. But if you put the spot arrays with the real detector efficiencies, it's good, but it's not that good. So, but technology will follow. So there's been many tracking works lately associated to MINFLUX. Originally 1 emitter in 2D, lately 2 meters in 2D with 1 single color or with 2 colors. We are doing 2 emitters in 3D with 2 colors, with all this arrangement of hardware that I showed you. So of course you could add even a blue one there, but it's already complex enough to do it with green and red. So with that library of beams, we can do conformational tracking. This means that we look at our single molecules there, we find them, then we put a feedback loop and localize in 3D with a set of exposures with a stop condition quite stringent. So once we're within 5 nanometers of the thing, we stop. And finally, we alternate between these 2 patterns to get traces. So the raw data looks like this. What you get is 14 traces, 7 for one color, 7 for the other color, as a function of time. And these traces you should turn into localizations by using some maximum likelihood estimation, which is also not trivial. It's not so easy to, when you're in the 1 nanometer, if you expect to measure distances with 1 nanometer errors, and you're 10 microns away from your origin, you need to keep 2 beams on top of each other, 1 nanometer in 10,000 nanometers. So this is, you need calibration precisions in 10 to the minus 4 to get this right. So this is not so easy, but we managed. What you see here is an origami that has 2 oscillating handles with dies. And here it has single handles that can bind any of them. So you can have the same die on the same location of the origami, to make sure we're actually doing this right. And okay, this is not the greatest example because this one flipped only once here, but we have more examples. This animation looked nice, that's why I'm showing this one. But the red one will appear here. All in all, the histograms are what speaks for themselves, so we can overlap them. There's some error, there's some bias, but this is 10 micros away, so you would need 10 to the minus 5 precision, and then you should go to nonlinear calibrations anymore. And this, it's enough, and it's good enough. That's the thing. So we are looking at cohesin. As I said, cohesin is this protein that is that has been shown to extrude DNA, and it's not clear how does it do this. So it has several binding sites, several ATP binding sites, but the mechanisms aren't clear. There are people studying this with cryo-EM, but this has the hurdle of, you need to lock your complex in position. You can do FRET, and it has been done, but you can look at pair of sites, and this is a very flexible thing. Here you have high-speed AFM, but high-speed AFM is, the substrates are weird, putting DNA there, not so easy. So all in all, this is a good thing for MINFLUX. We have initial preliminary data on this. So looking at the engagement of the two heads, they're labeled with SNAP and HALO. We have other sites, we have 5 sites labeled orthogonally with other systems as well. And the head engagement versus the ATP and other cofactors and DNA, it has been shown or is expected to increase. Now, the raw data looks like this. So we have a bunch of molecules. We pick the ones that are yellow, that have both of them. And then we track them. And we saw all sorts of things. So this is very rich. So the two partners are, very fast engaging with each other. Then they go together for a while. Then they stop. They go again. So there's a lot going on there. And I think it's going to be more challenging than we expected, but just because we found some, much richer variability than we expected. So what we have to do is measure more and get models and segmentations in place. Not even hidden Markov model would be not enough for this. So we need to go fancier. On the multiple, of course, this is a brute force approach, right? We have green and red, and but it's better if you can do this with lifetime and use a single wavelength. So you can have different dyes with different shifts, and if you expect whatever this ratio of channels for a dye, this ratio for the other, if they're both emitting simultaneously, you're going to get some ratio of spectral emission of your channels in between. So if you the same thing with lifetime, right? You're going to have some mixture, and there are if you look at the lifetime spectrum. There's you have channels there, and so the data would look like this. So you get for each one of your exposures, you get a lifetime and spectral histogram, and this is simulation, but we're testing, and it's okay. This is in 3D. This is ZX with realistic beams, realistic background that we showed that it works well, and this is for a dye being scanned around and the other dye standing in the middle. You can so this is localizable very precisely in 3D. So the next step for this is to do more molecules. So you do 3 and 4 and 5 and see where is the limit. At some point it will all break apart. But this has a challenge that is when you do maximum likelihood, you're estimating X, Y, Z, SBR for each molecule. So this becomes a search space of 4 dimensions, 8 dimensions, 12, 16. So this is not so easy as well. It's a landscape that is very rough. Now, at the end, I want to switch gears for DNA Paint. We've been working on this, and DNA Paint is interesting because of completeness, dye stability, kinetics control, like multiplexing. And MINFLUX and DNA Paint works. There's no problem. I think that the demo system of a barrier, actually, they demo it with DNA Paint strands. So why do they look the same? Because the storm, while storm MINFLUX traces look like this, DNA Paint traces in MINFLUX look like this, because you have floating dyes around, there's spikes everywhere, signature background ratio is bad. So I was very interested when this work by Kenny Chung at the Jörg Bebersdorf's lab came out, where they used fluorogenic DNA Paint. So they had a quencher and a dye, In solution, these are interacting and the fluorescence is quenched. And when this thing binds, they separate from each other and they control the kinetic with mismatches on the DNA strands. And I said, okay, let's try this. They are very fast, 20 milliseconds is a binding time. And we could see that, yeah, for wide field is not great. In our hands, this is the best we got for the, DNA origami that has 60 by 40 nanometers. But when you do MINFLUX on the same thing, well, it does well because it's a photon-starved situation. Yeah, photon-starved situation because it's fast, then you can gain something out of it. We also made our own DNA strands that were slower. And in wide field, this can perform well. And please keep in mind, this is 2 nanomolar, and this is 50 nanomolar, 25 times more dye. But still, the background, you cannot see in the screen, but the background here is even lower. The downside is that these have a lower K-on. But out of full curiosity, what appeared to be interesting for me is that, okay, can we create a library of these things? Can we create in the orthogonal imagers? But even more, can we create an in-between a handle that will bind to both of them? Because if that is true, we can have 2 imagers and 3 targets, 4 imagers, 2 imagers and 3 targets, 3 imagers, 7 and 7 targets, or 4 and 15. So you can scale exponentially the number of targets with number of imagers. And this might be interesting for MINFLUX and so on. So we took a very rational approach to this. We said, okay, let's think mathematically, how should the nucleotides be distributed? And what it turned out is that, yeah, we can distribute, for example, we can make a handle that has mismatches in red to 1 imager, but more mismatches another handle can have for another imager. So if you write it all down, you can go rational. So we put this, we randomize the nucleotides that would occupy these slots in NUPAC to predict most probable secondary structures or primary structures and binding energies. And we found that this rational scheme gives you a binary mapping of imagers to handles. So exactly what we want. So once we saw that, we said, okay, let's check if our imagers are orthogonal, which they were. And then we went a bit stronger. So we took against the imagers, we just evaluated the 67 million possible strands and got hundreds of candidates for each handle and measured the kinetics on 10 of them. We did this for all the handles. And finally, we got a very flat map of binding energies. And the kinetics and K-ons were decent. This has to be fluorogenic. So we made a library of fluorogenic strands for exchange with Cy3B and a quencher, but also for spectral and mixing with different red dyes and different quenchers. We chose the best ones. And we went for it. So what you see here is a DNA origami. So 6 origamis with the three strands. These are pretty well separable. What you see here is a ternary plot for the stoichiometry that you measure on each side of an origami. And these are the marginal histograms on the sides of the triangle. Confusion matrix is also great. And this looks a bit funky, but when we put this on nanobodies and do imaging directly on nuclear pores, looks great. So this is doable. Now, what we realized, okay, we can do this. We can do exchange paint, we can do spectral mixing, but also instead of doing this redistribution of mismatches on DNA, which is very interesting, we can also do this modularly. We have a strand that binds more than one thing. So we went for this as well. And you can concatenate binding sites. This has been shown by the human group, and it's a super work where they show how fast these strands can be. But if you mix them up, you can have, for example, predicted stoichiometries of 50% binding for each site. So we wrote down the likelihood function for classification and so on, and we can compute the probability of error of classification versus the number of events or binding events that you have on DNA Paint. We saw you can get to 1% error with 10 events or so, which is very generous. But also, you can go a bit crazy on the stoichiometry. So you can try to squeeze 28 targets on 3 imagers on sequential models of 5 or 6 targets. And it's interesting that you can have 9 targets, for example, with many very different distributions of stoichiometry. Some of them are very bad, some of them are much better. So you need less events to achieve the 1% classification error. So we did this for DNA origamis for 6 targets, and we did this for 9 targets, and this is working well. So the future for this is to go into DNA and RNA nanoscale tracing for the reasons that I showed you earlier. And will this need MINFLUX or not? Made in 3D, probably. Also, the nucleus, the idea is to look at this in the nucleus, very crowded space. So all in all, what I showed you is some technology of optics, some dual tracking of MINFLUX in 3D, a hint of the software, but we have a whole library of software for simulation, processing, and visualization. Hopefully this come out this year. And finally, our efforts on combinatorial DNA paint multiplexing. So that, I just want to mention that I am at IMP, which is a part of the Vienna Biocenter. It's a cluster with 140 groups, lots of topics and researchers. It's a great place that you should have in your map for sure. This is all I'm going to say. And with that, I want to acknowledge my group who's done all this amazing work on hardware, on computational stuff, on the biochemistry, and of course we have open positions and our funding sources, and that's it.
Anybody have any questions? I have a million questions, but
shoot.
Should I start? How fast do you run your feedback for stabilization? How fast does it have to be?
The acquisition, the cameras, I run them at 100 hertz. Mm-hmm. The update of the stage and the loop, I updated at 10 hertz.
And that's
so mild. Software based. It's all. The timings are not, FPGA timings. These are. So I acquire from the camera, I compute, and I send USB instructions to the stage. So this is a loop mediated by in the millisecond precisions. It will not be better. There's a right.
Very nice talk. So I'm wondering what kind of objective you are using and how deep you can image in the cells. Do you have any limitations with that? Yeah.
I'm using an immersion objective, 1.42 60x X series of Olympus, just for the field of view in case I want it. In Göttingen, we used like objectives of 140 something. I don't remember. It was a bit above 142. So anything will run well. These are not silicon oil objectives, meaning that so if you wanted to go deeper and have a good collection efficiency, you should do it with those ones. I don't expect any problem with that. I even expect less problem because at least you don't have, so with this, you have a mismatch of refractive index from the glass, oil glass to water, right? And the deeper you go, you get more and more spherical aberrations. Spherical aberrations will not degrade the zeros of the vortex of or the top hat, but it will make them larger, because these are beams that are, circular, symmetric. But with silicon oil, you will not have that problem, but the NA will be lower.
We have a question. Yeah.
Online.
Robert, go ahead.
Robert.
Yeah, sorry, I didn't find the raise hand function. Yeah, super impressive work, Francisco. It's really a whirlwind of cool advanced techniques. Actually, you had one point on one of the slides that struck me a bit, and I wanted to ask about it. So you said what is not necessary is fluorescence. But then, the way I understand MINFLUX, you still need to sort of, stochastically activate. Can you sort of explain or elaborate a little bit on this? Like, how can you achieve this without fluorescence?
Yes. So what I meant is that, going back to this point, I need the stochastic blinking if I want to image things that are close by to each other. I cannot separate them any other way. But the localization process itself. So we also do tracking, and other people too, with a single. There's no blinking, right? There's just a single stable emitting dye. So if you have a single scatterer and you do a sequential excitation, all the same principles apply, and the collection of scattering also follows Poissonian statistics. So all of this, it's like no step in the derivation falls apart.
Sure. Okay, I see. But then for, let's say, very dense samples, it would not work, right? Then you still need
No. Right. I understand. I understand why you're saying this. Now, of course, if you have now, what I think might be interesting is, well, I'm not very well versed on people doing tracking with iSCAT. I don't know if there have been attempts to merge it with structural elimination, but they should gain something out of that, in my opinion.
Well, thanks, but it's exciting thinking about, how you can maybe achieve similar precisions, right, in sort of non-fluorescent based.
Yeah, but we haven't done any experiment on this. Yeah, I want to stress that. But there's nothing preventing this. I think some people are doing confocal iSCAT, and those ones should just put a vortex plate that is nothing, and you get a very decent zero right away. And first, my opinion, they should first do the math and then do the experiment.
Yeah, very cool. Yeah, thanks a lot, Francisco.
Yeah.
Yes. Excuse me. You were critiquing the use of Fourier ring correlation, I think earlier, and I was wondering you could delve into that and other metrics, and how do you think the field should address these kind of comparisons?
This.
I think so. Yes.
Yeah. So, but I didn't hear the question.
Oh, yeah, I was. You mentioned Fourier ring correlation, and I think you had a bit of a critique about that.
No. Well, I can. I think that the Fourier ring correlation is a good metric as an entire curve. I don't think it's a good metric to just report when this curve crosses an arbitrary threshold. This is my opinion, because why? 1/7, fine. This is what people are doing, but I think all in all, the histograms or the OTFs or spectral distributions are what tell you something. So that's why I try to always show the histograms, not just the number, so you can see what this actually looks like. Also here, right? Because look at this histogram here and this one here. Okay, the median moved, but also the histogram it does look different from this one. It's not the same thing, right? So there are many of them who really suck, right?
Thank you.
You have a behind you.
It was a really great talk. So I'm curious, it would seem like this method could sort of bridge fluorescence with electron microscopy. Is there anything here that would sort of it would prevent you from thinking this could be adapted into a cryo scope where you could actually map the fluorescence with the level of precision where it would really sort of be attuned with the electron microscopy?
Yeah, that's a very good point. There's a lot to say about that. MINFLUX is very appropriate for scenarios where you're photon starved or Na starved. So if when you do you have a cryo fixed sample and you want to do light microscopy on it, you will use an objective that has a large working distance. Therefore, the Na will be lower, and then your collection of photons suffers, and your actual precision of localization, if you have molecules blinking, suffers. So MINFLUX is a good. There's a good justification to think of MINFLUX for that purpose, as well as structural elimination. To be honest, right? Structural elimination should be good. It gets you more. Now, what is the problem with this? On one hand, a lot of opinions. A lot of claim work has been driven by experts on EM, that are people who are more of an expert on EM than on light microscopy. In general, I'm generalizing. And therefore, the quality of the EM, data has been way higher than the quality of the light microscopy part with hurdles that it has. And therefore, at the end of the day, the interesting information came, I think, I'm sure there's some counterexample, came from the EM part and not from the light part, right? So many people in the field are jumping into this. There's an efforts. Some people are doing 4-pi cryo-SMLM. Some people are doing cryo-MINFLUX. Some people are doing regular SMLM. So, but my opinion is that whoever nails the blinking wins. And if you nail the blinking, so I think it's a chemistry problem. If you nail the blinking, you're going to get 1 million photons anyway, and you will not need, it will not be a photon-starved situation, even if it's an high NA and numerical aperture starved situation. So that's a reason why I didn't jump into this topic, because I'm not a chemist. And I think that the trick here is about the chemistry at cryo temperatures. It's a long answer.
We have a question online.
Hi, thanks. Really nice talk. I was wondering about, so I was really interested in the way you do multiplexing using the mismatched DNA strands. And you use, so the overlap of 2 different strands to get this multiplexing. And,
Yeah.
but you can also, is it possible to use like binding lifetimes to add, you know, complexity to this?
Oh, yeah. So, a couple of things about this. Do I have the slide for this? I don't have it. There's a lot no, I don't have it. So for each one of these, we measured the brightness of the fluorophores, the binding times, what else? The K-on. So all of these things differ. And if you, on top of the stoichiometry of which dye, which imager you see on each side, you add these sources of information, it helps a lot. Especially helps a lot for the case of the modular strands, because it's something that happens in DNA paint that perhaps is not the most advertised feature. Is that you may have, when you do exchange paint, you may have damage of the strands. So they do get damaged with radicals. And then you may do the round that does the black one, and then the strand is damaged and the blue one never binds. But still, from this black one, you expect, I don't know, 3-fold decay on because you have 3 copies. And from another one, you expect 1-fold. So the event rate will also help you distinguish stuff. So this all comes together, but we're not trying to predict this. We just see what the data looks like and help our classification algorithm just uses this information. But it is all there. Also, fluorescence lifetime, if you measure with lifetime, yes, because in the case of the quenching, but the other ones too, because regular DNA paint strands, short ones with a dye and no quencher, still have a fluorogenicity level of some of them 2-fold, and you see lifetime changes as well. So there's a lot of extra information around if you have it available.
Cool, thank you.
Great talk.
Thanks.
So it makes me think about a lot of things about what's going on with whatever the scatterer or the fluorophore is with regard to the excitation field. And so I heard you that it doesn't require single molecules, it's gradients. And so when I think of like dipole excitation, I'm thinking about, well, first off, in the molecules that you're talking about, there is dipoles that move around, right? They are not stationary, both absorption and emission dipoles. So I don't really see, I gather that you're averaging over that. But you would probably see an effect if you were to switch around your polarization of the light. That's the first observation. And the second one is, in dipole electric field interactions, there's actually an expansion of higher order terms, which is related to how sharply the electric field is varying in space. So if you try to make that gradient very sharp, you're going to have some, I don't know what the effect is. I don't know exactly what the magnitude is, but it's on the order of like a wavelength.
Yeah. No, you're onto. So this is all real. So I have a lot of things to say about this. Sure, if you, as you said, if your dipole is also like rotating Brownianly completely, sure, everything is nice and easy. If you have a dipole that is partially locked and is doing something like this in a cone or something, and there are people looking into that, people that are shaping, Matt Liu, he's shaping PSFs in a weird ways to extract, to distinguish, fixed dipoles at an angle from wobbling dipoles and so on. So all these effects are measurable. Now, something interesting that I don't have a good cartoon for, but this beam, I wish I had the whatever. So across the axial direction, this beam is polarized horizontally in X. Across the lateral direction, it's mostly Z. So when you actually do MINFLUX with 7 exposures of this, displacing laterally and displacing axially, when you displace axially, you're probing the Z component of the dipole. And when you move axially, you're probing the X component of the dipole. And if your thing is stationary, your photon counts are going to be informing on the orientation. And in this case, this would not be a feature. This would be a bug. Now, something that we're doing, an extra reason, but the thing is, I didn't sit down to test this experimentally. But as I showed you, we're mixing beam shapes. We're doing the lateral with this and the axial with this. And with by the way, this beam, the vortex beam, across the lateral direction, the electric field is lateral. So when you do the X, Y's with this and the Z with this, you probe the molecule's dipole with always X polarized or horizontally polarized electric fields. And therefore, problems with that will go away. Of course, if a dipole is fully vertical, I would not, so my brightness would be lower. But at least I don't mix in orientation with position, which is what happens when you probe with different sides of the beam and have different polarizations inherently. I have this beautiful animation somewhere, I cannot find it. So I think this is an issue that should be, so it should be experimentally tested how terrible or not it is, and when does it introduce bias on the localizations? Does it introduce bias on the localizations when you go below 10 nanometers, when you go below 5, when you go below 1, or when you go below 0.1? Where's the bound? When is this bad? This I don't know, but I know it's real and that this threshold will be somewhere, but I don't know where it is. This is my long answer. Very serious.
I guess there's no more questions. Thank Francisco again.
Thank you.